I welcome you all in organic reaction mechanism and biogenesis class. My name is Dr. R. Kale, working as an assistant professor at Department of Chemistry, CHEM College, Nasik. Students, in last lecture we have discussed about various methods to determine the reaction mechanism. In that context, we have discussed how the nature of the product is important in determining the reaction mechanism, and we have discussed some of the example also by which we can determine the reaction mechanism. In addition to this, we have discussed that how the intermediates important or the presence of intermediates play important role in determining the reaction mechanism. In first point, while determining the reaction mechanism from the nature of the product, I give you the example of Beckmann reaction, where acetophenone is treated with hydroxylamine and it gives this ketoxime derivative. In addition to this reaction gives this ketoxime isomer also. Subsequently, what happens? If you observe both the isomers, this hydroxy group is anti-periplanar to the phenyl group, and in this isomer, this hydroxy group is anti-periplanar to this methyl group. On subsequent reaction in acidic condition, I told you last time, whether it is a Bronsted acid or Lewis acid, it is going to rearrange and we will get this product. It is going to deliver two products, that is one product A and product B. Product A is N-phenyl acetamide and product B is N-methyl benzamide. Students, if you will clearly observe that reaction starts from the same intermediates, it gives the two isomer, and this two isomer gives rise to two different products. These different products clearly indicates that this reactions takes place with the different intermediates. As I told you that if this phenyl gets migrates, in one case we are going to get N-methyl acetamide, and when this methyl migrates, we are going to get this N-methyl benzamide. Next topic we have covered that determination of presence of intermediate. In next method we have discussed determination of presence of intermediate. If you remember, I gave you the example of Hoffman rearrangement. In that, amide has converted into primary amine in presence of NaOH and Br2. I have discussed this example also, where it forms the sodium hypobromide, and in this reaction, by isolating the different intermediates, one can determine the reaction mechanism. In addition to this, I have discussed one more technique by which one can determine the reaction mechanism, that is the physical methods. Physical method is nothing but the spectroscopic techniques like NMR, mass IR. This technique is particularly useful for the unstable intermediates. By characterizing that unstable intermediates, one can determine the reaction mechanism. Today we are going to discuss in similar context how we can trap different intermediates to determine the reaction mechanism. Students, when we talk about the carbocation, we know that order of carbocation stability. Carbocations are more stable in tertiary carbocations are more stable than secondary is more stable than primary carbocation. Suppose trapping of carbocations. How one can trap these carbocations to determine the reaction mechanism? We'll see an example. This unsymmetrical alkene on treatment with bromine, it gives these intermediates. As I told you that at the outset of this point, tertiary carbocation is more stable than secondary carbocation, and secondary carbocation is more stable than primary carbocation. So if you observe here, we have found secondary carbocation here. Subsequently, what happens? Another Bayer minus from the reaction, it makes the attack on this electrophilic center at this carbocation, and it gives dibromo compound. In similar fashion, if you treat this intermediate with various nucleophile, we can confirm that this reaction is working via carbocation intermediate. Suppose if we treat this carbocation with a hydroxide ion, we are going to get this OH. This OH makes the attack on this secondary carbocation, means this reaction has formed this carbocation. In next reaction, if you treat another nucleophile, cyanide nucleophile, in similar fashion, it is going to deliver this product also. If you treat another nucleophile, nitrate ester, again it is going to deliver this product. So all this reaction, what it indicates is that this reaction is going via carbocation intermediate, and by using these different nucleophiles, we can trap the carbocation intermediate and we can determine the reaction mechanism. Next method we will see trapping of free radicals. Trapping of free radicals. Students, these reactions are very simple, very basic. You might have studied in your undergraduate classes. Very classical example is that when we treat methane with a chlorine in presence of light, it gives this methyl chloride. So generally what we talk about, as you have studied in your earlier class, when we see this photolytic condition, we generally say this reaction is working via radical mechanism. But question raised here that how to determine or how to identify whether reaction, or how to confirm that whether reaction is going via free radical pathway or not. So by treating free radical with different trapping reagents, we can confirm that this reaction is working via free radical mechanism and not by cationic mechanism. For this purpose, if we treat this reaction with this nitroso compound, in this reaction it gives it generates this nitroxide radical. Students, keep in mind that nitroxide radicals are stable and we can characterize it. Once this compound has formed in the reaction, this confirms that this reaction is working via radical mechanism. Let's see how this reaction takes place in radical way. As I am writing, keep writing you also. First of all, it induces the radical formation. It gives this two biradical spaces. Subsequently, one chlorine radical abstracts this hydrogen, it generates or it induces the formation of radical from the methane. This methyl radical forms here. Then this methyl radical induces another radical from the chlorine. It forms this methyl chloride. Again this chlorine radical will take part in the reaction and it will keep continuing this reaction. But if you remember, I have shown this product here with this nitroso compound. So what happens when this reaction goes? If you treat with this nitroso compound, as I told you, it induces the formation of radical, and this radical this nitroso radical forms, and this nitrogen radical, for my convenience I am writing here, you just note it. It forms this bond and at the same time this reaction gives so students, by trapping this radical with nitroso compound, as I told you that it forms this nitroxide radical and it confirms that this reaction is working via free radical mechanism. Another interesting example we will see. These are all basic reactions, but you know, it's interesting reactions. Another classical example we will see, that chlorination of toluene. Chlorination of toluene, again in presence of light, it gives this chloro product, but at the same time if you trap this reaction with a nitroso compound, we are going to get this nitroxide compound. So in similar fashion this reaction will also work. Radical radical will get induced, then another radical will form from the neutral molecule, and then subsequently that neutral molecule radical will react with this nitrous compound and will deliver this product. So student, now what you want to do is that, as I have shown the mechanism, you write the mechanism for this reaction. Next topic, trapping of carbene. How to identify or how to confirm the formation of the carbene in the reaction. To understand this, we will see this reaction. Usually carbenes are trapped by alkene derivatives. Alkenes are used to trap the carbene. If we'll treat the cyclohexene with chloroform on heating in alcoholic KOH, reaction will give this cyclopropane derivative. So now how to identify that whether carbene has formed in the reaction or not. So I hope you might have studied this reaction also in your TY basic class, generation of carbene. That is very famous reaction. After this reaction I will tell you what is that reaction. Treatment of this chloroform. Base I have shown here. This base will abstract the proton. It will generate this negative charge on this carbon. Subsequently, on alpha elimination, will give this carbene, and then subsequently this carbene gets combined and will get this cyclopropane derivative. How it will get combined? It will combine by simple 1,2 addition product or 1,2 addition mechanism. For my convenience, I am showing here, you write it properly. Or I will write it separately here. This pi bond gets shifts, again that negative charge makes the attack on another carbon of this cyclohexene, and we will get this cyclopropane derivative. As I told you that outside of this example, there is another classical example you have studied in your TY basic class, that is a Reamer-Tiemann reaction. This is very famous reaction. What is Reamer-Tiemann reaction? In Reamer-Tiemann reaction, we treat phenol using the similar condition, chloroform or heating in alcoholic KOH, and it gives salicylaldehyde. Phenol on treatment with chloroform heating alcoholic KOH forms this aldehyd salicylaldehyde. So now what I wanted to know that you write the mechanism for this reaction and see how carbene is involved and how carbene is trapped in this reaction to get this aldehyde. The next method we are going to see, that is a trapping of nitrene. How to trap the nitrene? Students, keep in mind that whatever reactions so far I have discussed, by which we can trap the intermediates and we can confirm that this reaction is going via this intermediate, or this intermediate has formed in the reaction to get that product. Means we are getting cyclopropane means by treating chloroform, and in basic condition means reaction is going via carbene mechanism only. That you keep in mind, that will be useful for your competitive examination and your university exam also. So trapping of nitrene, as we have discussed that trapping of carbocation, trapping of free radical, trapping of carbene, we have used different derivatives, different functional group to trap the carbenes. Unlike these intermediates, nitrene is trapped with benzene. Nitrenes are trapped with benzene. So let's see how this benzene will trap this nitrene. Treatment of nitrene with benzene. Will deliver this cyclic product. Subsequently, on different condition, with acidic condition, it may give expanded ring expansion product, or it may give aniline substitute product. As I told you that in acidic condition, as we have moved this bond, so positive charge will generate on this carbon. So positive charge I have shown here. Subsequently, aromatization will take place. And it will form this substituted aniline derivatives. In another case, this ring may expand, or this ring may cleave. On expansion, this intermediate gives this product also, that is nothing but azepine. So nitrene is trapped with benzene, it forms this bicyclic system. It may deliver two products with two different conditions. In acidic condition, abstraction of proton, and subsequently aromatization will give this substitute aniline. In another case, it may deliver the on expansion azepine derivative. The next intermediate trapping we will see, that is a trapping of benzyne. Students, benzyne basically gives Diels-Alder reaction. How to form the benzyne? If you treat chlorobenzene with a strong base sodamide, it abstracts this adjacent proton and it develops this negative charge on this benzene ring. Subsequently, on conjugation, on shifting this electron, chlorine get expelled from this one, and it gives this benzyne. Intermediate. When benzyne intermediate treated with anthracene, benzyne acts as a dienophile in this reaction, and this acts as a diene. You keep writing what I am writing here. And it gives this tryptophan product. This tryptophan is nothing but this is a tryptophan. In similar fashion, when benzyne is treated with furan, reaction again works in a similar fashion. Keep it in mind. For my convenience, I am writing like this. You write it separate. I am using similar benzyne. So benzyne, when treated with anthracene, it gives this tryptophan. When this benzyne is treated with furan, this Diels-Alder reaction will deliver this product. So this reaction confirms that this product confirms that this reaction is working via benzyne intermediate. That's why it is giving this Diels-Alder product, tryptophan, and this furan tricyclic system product. The next reaction in this context we will discuss when benzyne is treated with furan, this Diels-Alder reaction also gives this cyclization product, that is nothing but so formation of these two products confirms that this reaction works via benzyne intermediate. Students, solve the given MCQ and other questions on LMS. In next lecture, we are going to discuss other methods by which one can determine the mechanism. In that, we will discuss the cross-4 experiments, isotope labeling, stereochemical studies, and kinetic evidences. By these methods, how one can determine the reaction mechanism. I am expecting that after completing this topic, you must be able to write the mechanism and you must be able to design the experiment for determining the reaction mechanism.